Hi, I'm here with my husband, Dr. Bill Kurtin, who is a biochemist. Most of you probably know Bill and his career at Trinity. Like most families, we've been talking about the persistence of the coronavirus. And the other day, we were talking about a new approach to vaccine. He was telling me about this, and it was really exciting. So I asked him if he would talk about it a little bit. It's actually a bit of a bright spot, and it's also very interesting. So, Bill, take it away.
Okay, well, I'm sure all of you are aware that there are lots of companies that are in a race to develop vaccines against this coronavirus and help us get over this pandemic. And many of the companies are using traditional vaccine approaches, but there are some new approaches out there that seem very exciting and interesting to me, and I thought I would talk about those. But in order to do that, I thought it would be helpful to talk about, first of all, how traditional vaccines are made, and that will help you understand how the new approach works. So I'm going to use a couple of visuals here. This one is just a picture. One particular image of a coronavirus, which you've seen many images of these things, characterized by these blobs or spikes on the surface that are referred to as the spike protein. And it's that spike protein that your immune system makes antibodies to interact with. So those antibody proteins will attach to these spike proteins and identify the virus to be taken care of. So in making a traditional vaccine, what companies have to do is first of all they have to grow up a large amount of these virus particles, these intact virus particles. So they isolate these virus particles, purify them, and then they have to weaken the virus particles or inactivate them using chemical methods, and there's a variety of different methods they can use to do that. But once the virus has been weakened, then they isolate those weakened or inactivated particles, put them into a vaccine, and inject them into you. And because they are weakened, they are not supposed to induce any kind of disease, although that does sometimes happen. But again, what happens is your immune system sees those particles, responds to the virus, the surface proteins, the spike proteins, and makes antibodies. So hopefully the immune system will remember that, and you will basically be immune to the virus in the future. So that's the traditional vaccine. Now, an alternative approach simply says, what if we can just somehow get normal cells to make this surface protein, the spike protein? And if they will do that, then again, your immune system would recognize that as a foreign protein and make antibodies against it. So in order to figure out how to do this, or to demonstrate how to do this, we need to look at how cells normally make any particular protein. And so I have a second graphic here, this is just a cell with a nucleus in which you will find the DNA, like you would in any normal cell. And that DNA has all the information in it to make all the thousands of different proteins that you'll find in your cells. So the information for a particular protein is called a gene. And so what happens in a normal cell is that if we're going to make a particular protein, in the nucleus, the information in that particular gene is transcribed into a smaller nucleic acid molecule called an mRNA, which just stands for messenger RNA. And then that messenger RNA goes out of the nucleus into the other part of the cell. It's called a cytosol. And there's a lot of molecular machinery there that takes that encoded information in the messenger RNA and uses that information to make the protein of interest. So that's the normal sequence of events that occurs in a cell to make a protein. You start with a gene and DNA, you make a messenger RNA, it goes out, and then you use that information to make a protein. So the idea is, if we want this protein to be the spike protein, all we have to know is what is the genetic information, what's the gene for making that protein. And from that, if we can make an mRNA molecule that has that information encoded in it, then this molecular machinery in the cell can make that protein. Once the protein is made, it will be, again, recognized as a foreign protein, and your immune system will mount a response to that. So what one particular company is actually doing is making these mRNA molecules. You can do this. There are biotechnology methods that enable you to do that once you know the gene sequence. So you make the mRNA, you inject it into a person, and the cell starts making the protein. It mounts an immune response against that protein, and if all of that works the way it's supposed to, it should be very effective. And the advantage of that is that it is much simpler to make these mRNA molecules. They can be produced very quickly, and then once and therefore they can be made less expensively and widely distributed very easily. So you're avoiding all of the business of trying to make intact virus particles and isolate them and weaken them and inactivate them and so on and so forth. So this is all quite exciting, but as you might expect, there are problems to overcome regarding whether your mRNA molecule is going to cause some unforeseen immune response that is undesirable, and things like that, how to get the mRNA into the cell, and so on and so forth. But there is actually one company that's got this to phase 3 clinical trials. So it's really quite exciting. Another company is actually using a method whereby they're putting a small circular DNA plasmid, called a plasmid, into the cell, and then that circular DNA makes, from that, an mRNA is made, and then that goes on to the spike protein. So there's a second company that's working on that approach, which involves starting with DNA instead of the mRNA. But everybody's really excited about the possibility that that's going to work, because if it does, again, I think it will mean that we'll get a vaccine more quickly and less expensively than using the traditional vaccine approach. So that's about it, and I'm just cautiously optimistic that this is going to work.
This is really good news. As we follow along with the production and the testing of vaccines, what should we look for when we're looking for progress on this subject?
I think it's going to be mainly a question of how far, what has happened in the clinical trials. They have been fairly small scale, and so those trials have to be scaled up to answer the very basic questions concerning the safety of the vaccine for humans. So that's what we need to watch for.
Well, if scientists are optimistic, that really is good news for all of us.
Right.
Thank you for explaining it, Bill.
You're welcome.